thing actually is going to address some of the things that we've been talking about, but try and actually dig a little bit deeper on perhaps common mistakes that people make when they're looking at different kinds of samples or interpreting the data that they have. And it's going to come back to some of the stuff that we're talking about is exactly, how much do you have to think about the provenance of samples that you get, how the samples are collected, and perturbing factors. Okay, so I don't have too many slides because I wanted to keep this open for, and this is going to be more interactive because I'm going to ask people to guess about, what things went wrong. All right, and hopefully I won't drop everything this time. All right, so this is a general thing about sample collection and data experimental design, and the things that we've come across, and this is really looking at literature, reviewing papers, mistakes that we've made, mistakes that other people have made in designing and conducting the experiments. And it gives you an idea of the breadth of things that you'll need to think about. So one of the things that is also that's really important, and this is where we're starting to kick out experiments, is the biological plausibility of a metabolite giving you a biomarker of something that you're interested in. All right, and this is really, it's a really important thing because people want to do metabolomics on a lot of different things. And no doubt that metabolomics, because of what we've been talking about, that they're very sensitive to environmental perturbations, they can be used for a lot of things. But sometimes I think people are asking questions that just aren't going to be able to be answered by metabolomic profiling of especially human subjects. Okay, the biological plausibility that the response we've seen in the system being studied in rodents versus humans. Now, we talked about this already, is that rodents, remember, if you do a mouse experiment, you do a human experiment, mice, turns out, have a higher metabolic rate. They eat a lot differently than humans do. They have somewhat of a different circadian rhythm for a lot of different metabolic processes. And so if you get a result in a critter, it may not be directly transferable to humans, and so when you design the experiment based on human physiology versus a rodent physiology, you need to take this into account. And there's certain things that humans can do, that rodents can do, that humans can't do, and certain, the microbiome is different. There's a lot of differences. So just, make sure that you want to make that you don't extrapolate too much. Of course, we all use rodents to try and first test our system, but for instance, lecithin knows, there's a malic enzyme in beta cells that's absent from rodents, and it actually changes the intermediary metabolism that you see. The black 6 mouse, the C57 black 6J mouse, has a specific mutation in a mitochondrial enzyme that changes their mitochondrial metabolism. And so if you're looking at at the details of those kinds of things, you need to make sure that you understand the model that you're working in. Okay. There's the biggest thing is extraneous variations in phenotypic endpoints that really muddle the relationship to the metabolome. And I'm going to show you a couple examples of this that are sometimes pretty obvious, and sometimes you have to dig a little bit to really understand that. And this is the how many factors can affect things is infinite. It doesn't mean that you can't find things, but you just need to make sure that you think about certain things before you do an experiment. One of the important things when people come and they want to do an experiment with frozen samples that may have been thawed or something like that, is how do you know when you actually have a result? It seems kind of obvious, but a lot of times people are coming to us and want to do experiments, and my challenge to them, if you're going to look and see if a person who gets sick on this compound when they take this drug or have this reaction, how do you know when you actually have a biologically relevant result versus a spurious result? Especially if you're doing untargeted metabolomics, you're doing large sample number or large metabolites, and your N of the individuals that you're testing, and for in most cases, is a small number. And so to be able to understand these differences, unless you can do it paired, you can do interventions before and after, and you can actually see changes in individuals, just cross-sectionally, it's very difficult to do that. And it's really an important thing to remember, is that we're going to give you a lot of data. It's up to you to figure out what's going on, because you're the content expert. So when you find those metabolomic profile differences, we need to understand. Ala and I were talking earlier, is that hopefully we can develop methods to really look at the relationships of large numbers of metabolites and not specific metabolites that may help us understand sort of the biological implications of some of the interventions that we do. Power calculations, we've talked about this a couple times already this morning. I won't go into that, but knowing the error in the measurements is one thing, but knowing the error in the collection time, all these different things, usually you need more than you think you do. And then I'll show you an example of how not to design an experiment with multiple groups. All right. So first thing is that the biological plausibility that metabolomics response was seen. So this is an experiment, and I'm trying to, I don't think the person who did that is in the room. I'm trying to want to make sure that when you design an experiment and you collect samples and you spend a lot of time collecting samples and then you run a metabolome, it's really good to look at your biological perturbant or your phenotypic endpoint that you're looking to see if there's a relationship between, to make sure that there's something going on with this. So this is an exposure of one type or another, and there was multiple exposures measured in these samples in a number of adolescents, over 100 individuals. And exposure 1, exposure 2, you can see there's some variability here. You got an outlier here, there's up and down. And here's phenotype 1. Obviously, this is systolic blood pressure. Here's phenotype 2 is triglycerides. And there was some question whether there was going to be a relationship between the exposure and the response. The big problem is that this is the normal level of each one of these the upper limit of normal of each one of these either phenotypic endpoints or exposure. So what you see is that just about every sample is within the normal range of exposure that's been determined, and just about every measurement of the phenotypic endpoint is in the normal range. And so you don't have any outliers, you don't have a lot of variation, because the normal range tends to constrict your dynamic range. Now, you could see if there's a relationship between normal levels of one thing and normal levels of another thing. But if you're looking for pathology, which this investigator was looking for pathology, and there's no pathology and there's no exposure, I dare say this is going to be a difficult experiment to publish at some point. And unless our statisticians can work magic, I think this may have been an experiment that gone awry. The biggest problem with this experiment is that these samples were submitted to us to do metabolomic profiling before the exposure levels were back, and the investigator was quite surprised that all the exposure levels were in the normal range. So this is a way that you can get into trouble, or a way that you can get poorer in your bank account, but we get paid. But it would be better if you, because now the investigator wants us to find something in his data. All right. This is from a paper that I reviewed, and it was really an interesting sort of paper. This investigator was assessing the effect of a drug treatment on fatty acid profiles in the plasma of men with high-risk cardiovascular disease. And so they treated some men with a statin, a drug intervention, and the other ones were left as controls. They were identified as individuals in the database they had, and they had blood samples, and so they actually collected the blood. And so they said, wow, this drug that we had, this drug that we gave this individual, caused an increase in all these polyunsaturated fatty acids. And at least for a while, there was some evidence that increased polyunsaturated fatty acid was better for you as far as cardiovascular risk. That's sort of gone away with recent studies. So anybody want to guess what happened in this study? Why? So when I asked the investigator to look at something, he came back and went, okay. So this is a drug study. So the problem was that the controls were controls they identified in a database, and the controls were never talked to or entered into the study. The other group, the group that got the drug treatment, got the drug treatment. But it turns out that the drug treatment was given to the individuals, but the physicians who were taking care of them also did standard of care, which was give them omega-3 and omega-6 fatty acids. And I dare say that this change is due to the additional drug treatment that was given to these individuals that wasn't given to the control group, and so therefore you didn't understand that. So these are sort of things that happens when people design experiments, especially if you're not intervening in the control group. And this happens a lot. People are saying, well, I'm just going to take random matched samples to the people I'm doing these interventions in, and then didn't have the proper control and treating everybody the same way. So when you design your experiment, think about what's going to happen in your controls as well as happening in the intervention group. All right, so this is one of my favorite ones, simply because we were working on something similar to this group, and we're quite upset that they got a very high impact paper out of this, although we were not getting the same results. And so this is, again, these are samples that were derived from archival samples. So individuals had done experiments from different institutions. Somebody's talking about different institutions and how samples are collected and how experiments are going. And so, this group actually was able to publish this paper. And basically what they said is that this is in relationship to branched-chain amino acids and whether they cause insulin resistance. They say the concentrations of several individual plasma amino acids decreased significantly after gastric bypass, this is bariatric surgery, versus the group that got a medical intervention, medical weight loss. And so this is the drop in the branched-chain amino acids, the total concentration of leucine, isoleucine, and valine. That dropped in gastric bypass, but there was no significant differences in the diet group. Despite that there was a tendency to go down a little bit, it wasn't there. And this is from two different institutions, from New York Medical Center and Duke cohort. There was a little bit of decrease there. So it was concerning to us because we actually had seen some reasonable drops in amino acids on people who are getting a very low-calorie diet. And so this is a normal, these are people who, after weight loss, a very low-calorie diet with about a 15% weight loss. And what we could see is that there were statistically significant decreases in valine and these aromatic amino acids and leucines in here somewhere. And we do get a reasonably good, 15-20% drop in there. When we actually have people on the very low-calorie diet that we use, which is a high-protein, very low-calorie diet, and we test them on that, actually we get an improvement in insulin sensitivity because they lose a lot of weight, their insulin and glucose levels go down. But actually it turns out these branched-chain amino acid levels are not different because the diet itself has a very high amount of these amino acids in there. It's a whey hydrolysis. So we started going back, and we went back a couple papers because when we looked at the provenance of the samples that were collected, the diet-induced weight loss, patients were kept on the 1,000-calorie diet, and they actually used the same kind of diet that we did, in negative energy balance. They were in active weight loss while they were retested for incretin levels. Well, this was the original paper that was used to use the same sample. The Roux-en-Y gastric bypass protocol, these people were given any diet that they wanted. So what we think is happening is that they are comparing apples and oranges. These people are on a very high protein, calorically restricted diet and active weight loss, whereas these people were about 6 months to 1 year after a gastric bypass, and they were eating anything they want. So again, it comes back to this diet that we're talking about. Long-term dietary interventions and short-term dietary interventions can make a big difference, especially if you're doing human studies. That these things, they do change, they're very sensitive to diet, and so really need to think about it. But also when you review papers and there's something funny going on, you just might want to, if it doesn't quite make sense, maybe it doesn't quite make sense. Okay. All right. This is a study from actually a really good friend of mine. I actually tried to talk him out of doing the study, but he had the samples that were in the freezer, and they decided that they want to run them. And so they investigating men and women, and the women with and without polycystic ovary disease, with and without sleep apnea during fasting or during slow wave sleep. Okay, now that's a lot of groups. Okay, and it turns out that there's not that many individuals in each group. And so if you do, this is a random forest or PSLD, I forget exactly what it was. Help me. That you can get some separation of these groups, that they're all clustered. And if you look at the metabolites that are driving the differences in each one of these groups, you can actually see patterns. Well, here's fasting, slow wave sleep, fasting, slow wave sleep, fasting, slow wave sleep. And this is phenolactone, which is part of the fatty acid intermediate breakdown. And it turns out that there's a nice pattern there. But the problem is that this is not the difference of effect of slow wave sleep. It's effect of fasting, because slow wave sleep was taken about, they had a snack before they went to bed, slow wave sleep was about 3 hours later, and then they were fasting in the morning. And what you actually see is the effect of fasting, not of slow wave sleep. But they wanted to publish this, that slow wave sleep actually decreases the level of mevanolactone in the blood, when it's clearly, it's really due to the sleeping patterns. So again, thinking about the data that you have in hand. Now, if you try and dissect other metabolites that are separating slow wave sleep in this group and that group, you can't find anything that really changes. What you can find is a fasting, non-fasting differences between the individuals. But all the other groups, because there's such small numbers in all these groups, that you can't find anything different in all of them. So again, it's the N that you're doing and how many comparisons you're trying to do. I dare say this is the most subgroups we've ever had in a human experiment. And again, these guys spent about $30,000 doing this experiment, even though I really suggested that they not do it. Okay. And this is one that it's still confusing to us, and we're waiting for the investigator to come back, but I talked to him, I've said I was going to show it to him while he was investigating. Investigator provided 100 plus plasma samples for lipidomic profiling, stating that a subset of subjects submitted were from samples from subjects that were sampled twice, about 10 years apart. And he wanted to see what the consistency of the metabolome was, or differences in the lipidome, was for 10 years apart. And so our ace statistician Tanu was trying to figure out what's going on with this, and it just didn't quite make sense. And then so what we did is we actually just did a simple thing. We plotted the age differences in the individuals with the same subject ID, and this is the difference. And so there was anywhere from 25 to no years difference between these, and when it was supposed to be 10 years apart. We think that they actually mislabeled these samples. And unless they can dissect this, we just spent $50,000 to do something that probably is not going to provide benefits. So and Kelly talked about it, label those samples, make sure the provenance of the samples are there. It's really, really critically important that you don't make these kind of mistakes. And this guy is a world-famous researcher. He thinks that somebody else did it. In fact, we've been working with this guy for a while, and this is some samples from a population in Texas that we actually looked at people that were actually a cross-section of people that were sampled in 1985 to 87, and then about 15 years later, and we ran these samples in 2013. So these samples were at least 10 years old. These samples are 25 years old. And we wanted to see how good the samples were. And this is somebody was asking before, how long does it take to go bad? So we know the relationship of a lot of different metabolites with a lot of different metabolites. And this is one way that we look at data And I'll show some more of this tomorrow on how you can look at the data. But we know that there's certain things. So this is the clinical triglyceride concentration that was done at the time of blood collection, either in 85 or 2002. And then we looked at the triglyceride fatty acid concentration. And what we find is a nice linear relationship between the two, which gives you an idea that, you know, what we're measuring is probably close to what was sampled there. And then we can look at specific metabolites that we know have high correlations with each other, acylcarnitines, which are notorious for being very stable. These are things that are measured in blood spots for perinatal diagnostic screening for inborn errors of metabolism. And so acylcarnitines will stay around forever, and these were done a long time ago. And we know that the acylcarnitine, which is generated from leucine, there's a really nice high correlation between the acylcarnitine levels and the leucine levels in the blood samples here, which makes us feel pretty good. And we looked at a lot of other things. What we find is that glutamine, glutamate, asparagine, aspartate, those are susceptible to changes over periods of time. And so those are the kinds of things that you need to worry about. And maybe one way to look and see how good your samples are. How much of a variation are there in the relative relationships of those. The amino groups tend to fall off. The terminal amino group in the double aminated amino acids tend to be labile. What was interesting about this is that if you look at the patterns, and I'll go through this, the patterns of in 1985 to 87, we've looked at some relationship between some targeted metabolomics, acylcarnitines, amino acids, and these fatty acids. And what you can see is that there's a much more modeled pattern here than there are here. You can see there's a higher correlation between a lot of these different amino acids and acylcarnitines. And what we actually found is that this is a pattern that's consistent with a high carbohydrate diet. And it turns out that the population here in 1985, there was one McDonald's in the main street of town, and it was a traditional diet. And this correlates with exactly with the number of fast food restaurants in the population. And believe that what we're trying to do now with these samples is actually look at the prediction of type 2 diabetes in the sample and correlate with specific macronutrient intakes, because the prevalence of diabetes here was about 11%. The prevalence of diabetes now here is 38% in the population. So it's almost 4 times as high. And so we're very interested to try, because there's samples throughout the years, and as the prevalence changes, we want to see if this dietary intake and what part of the diet is actually worth it. So I'm just showing you that you can use very old samples. He knows for a fact that his freezer hasn't gone down. Then there's something that happened in these experiments, and this is our sort of our mistake. So this is we're looking at fluxomics samples experiment, looking at the labeling of citric acid in macrophages. And we performed these experiments for a couple of different investigators. And a lot of people are looking at macrophage metabolism. There's some really interesting things related to inflammation and things like that. And so we did this for one person, and then we did this, wild type versus treated, wild type versus treated. This is treated with lipopolysaccharide. And here we got about 50%, after 1 hour of labeling, about 50% of the citrate is labeled when we give C13 glucose. And here, only about 4 to 5% is labeled. All right, so anybody want to guess? Why would there be such a big difference? Actually, everything was exactly the same except for the media was different that was done in. And the media that we actually gave to this person, turns out it had 2 millimolar pyruvate in it. And we just didn't, there was an oversight. But this doesn't have pyruvate, and pyruvate, which actually is the thing that ultimately labels the Tca cycle, was cold, unlabeled pyruvate, and was just inhibiting the labeling by the glucose, because pyruvate can go into cells and actually goes directly into the mitochondria. So again, it tells you about the conditions that you need to use to do these kind of experiments. I couldn't find the study, but it's also very, very critical. Did I put it in here? Yeah, I'll come back to this. So the other thing is that a common mistake that people make is when they're doing tissue cultures is that they want to do an intervention, and so they'll take and they want to add their drug, and then do an experiment after 15 minutes of adding the drug and seeing what the metabolism difference is. But commonly, the mistake that's made is that they just add the drug and then add the label, or just add the drug and looking at the metabolite differences. And when they add the drug, they refresh the media. And so very commonly, people keep a dish overnight, they wash the media out, and they add their drug to the new media. And what you find is that the new media is actually replenishing the depleted metabolites. And so you want to really make sure that when you treat tissue culture cells, you do exactly the same thing to the control cultures as intervention cultures. It seems obvious, but it's really, really amazing how many times this happens, that people are actually giving us cells where they weren't treated identically. In general, when we do fluxomics experiments, we suggest that people wash their cells from the media after their overnight incubation in the morning, feed them for anywhere from 15 minutes to an hour with fresh media to replete all the metabolite levels, wash that media out, and then add label for doing fluxomics experiments, trying to get the cells at least to be in some semblance of the same metabolic state when you start the experiment. So it's really important that you think about exactly what might be happening, because some cells have really fast metabolism, especially if you're having a treatment that's decreasing or increasing the growth of your cells. You have to think about the depletion of the metabolites in the media when you're actually doing the experiment. And so I just want to caution you about the intracellular concentration can go down and things like that. So that's another thing. And then interpretation of the data. So this is from a paper that Matt published a couple of years ago, looking at the concentration of different metabolites in a beta cell line after 5 or 10 minutes of adding glucose to the culture. So glucose stimulates insulin release. Wanted to see what the metabolite changes are very quickly, because insulin gets released very fast. So glucose comes in, goes into Tca cycle. The Tca cycle is very important for insulin secretion. Citric acid goes up, one would expect. Glutamic acid goes down a little bit, then goes back up. Malate goes up markedly during this time, and this is part of the Tca cycle over here, so sort of makes sense. Aspartate goes down, goes back up, and so it's like, okay, so malate goes up. It's really important, but this is just when you do unlabeled, or basically total concentration. If you look at the labeling, however, it's really interesting that citric acid, about 50% of it at 10 minutes gets labeled, if you don't have pyruvate in the media. Glutamic acid is labeled a little bit, but not quite as much, suggesting that a lot of the citric acid is actually exiting the cell. But if you look at the malate, it's actually not labeled much at all, and most of the rise in the malate is actually unlabeled malate. And so malate's not going up because you're going through the Tca cycle and increasing the concentration of the metabolites from the Tca cycle. Indeed, what we actually found that this probably comes to be aspartic acid, actually through the aspartate malate shuttle. And so there's actually unlabeled substrate coming in and supplying the malic acid from that way. And this actually points out that flux experiments can be very helpful in interpreting where these metabolites are coming from. And so if you do specific experiments, you can actually use specific labels to really understand how things are going in and out of different pathways. So it's really important to think about that. Oh, and this is an experiment I thought I was going to show earlier. This is the extraneous factors affecting metabolites. So this is static levels of metabolites and then flux experiments. And so the cells were fed for 24 hours previously, washed and harvested at time 0, and then at time 30 minutes after addition of 10 millimolar uniformly labeled glucose. And B, the cells were fed 24 hours previously, washed and refed for 30 minutes in each one, and harvested at time 0 or afterwards. And what you can see is that if you just did it this static incubations here, you can see that there's a whole heck of a lot more metabolites in the cells after a fresh refeeding than there were simply because they got depleted overnight. They were actually really high metabolic rate cells. And then if you looked at the amount of labeling that you get in the patterns, they are actually different, and you'll get a different answer depending upon how you look at the data. Okay, so it's really important to understand that you need to get everybody the same. And so this is the 0 time, and this is the after addition, and you can see that they are much more similar. You're just getting a rate of labeling, whereas the other one, you're getting the rate of reconstituting label and glucose in the cell. Okay, so be careful. Be careful, be careful. Be aware that things change. Be aware that target-on-target metabolomics give you a snapshot in time, so you can't say anything about changes and stuff. You can only do cross-sectional studies. And check and recheck your samples. Just make sure you don't waste money, because it's not cheap to do these experiments, and it's not cheap to work up these experiments. It's not cheap to collect the things. And so just talk to us. We can be very helpful sometimes. All right, I'll stop there. Any questions? Oh, thank you, Taiwan. My friend, he's always there. Any questions? I know it's getting a little late. Okay, so we'll all be around at lunchtime. Are we having lunch here? Terry, what are we doing? There's lunch outside, and